So today we will start from the next part of the continuing chapter, Cell the Unit of Life. Today is the lecture number 4. Welcome to Pre-Med Pro, and so we're going to start our new lecture, which will start from ribosome. So we will start it from ribosome. See, ribosome are of two types. One is 70S and another is 80S. So what does this S means? S means Sydberg unit, after the name of the scientist who has discovered it, Sydberg. Sometimes we call it as sedimentary coefficient. So what is the sedimentary coefficient? See, if you take a groundnut and crush it into fine pieces as well as some broken large pieces, and if you stir it or centrifuge it into a milk, so the large groundnut pieces will sediment at the bottom and the fine pieces will float on the top part. So with this we call it as sedimentary coefficient. Sedimentary coefficient means the large parts are generally deposited at the bottom and the loose parts are, or the light parts are generally floats on the top of the surface of that ribosome. Okay, so in prokaryotes we see as 70S ribosome, and in eukaryotes we see 80S ribosome. In prokaryotes, the 70S ribosome can be broken into 30S and 50S. 30S and 50S. So what does that 30S and 50S means? 30S is the lighter part, which is generally compared with the fine groundnuts, and the 50S is the larger part which gets deposited at the bottom. So we write this point, ribosome are 70S, which is broken into 30S plus 50S, and 80S in 40S and 60S. So S means Sydberg unit, and 70S seen in prokaryotes, and 80S are seen in eukaryotes. Okay, so in the next let me draw a ribosome. Okay, so now these are the ribosomes which are getting attached with the cell membrane. Okay, this ribosome gets attached with the cell membrane and this one is the ribosome which are at the center. Okay, this one is the ribosome gets attached at the cell membrane, the red one, and this blue one is at the center. Okay, now let me write ribosome attached to cell membrane, and this is a cell membrane, and this is the cytoplasm. Okay, in 70S, small subunit and large subunit. Small subunit and large subunit. So small subunit is 30S, large subunit is 50S. Okay, S is what I have said. What is S? S is the Seeberg unit. Seeberg unit. Okay, now the sedimentary coefficient. What is sediment coefficient? Sediment coefficient indirectly measures the size and the density. It indirectly measures the size as well as the density. So I can write as indirectly measures the size and density. Heavier particle sediments faster. The heavier the particle, it sediments faster. Heavier particle sediments faster. Okay. Note: DNA to mRNA to protein. Okay. Translation. By the help of ribosome. Okay. Now let me write. This one is a ribosome. This one is also a ribosome. And this one is also a ribosome. When multiple ribosome attach together, what they forms? Polysome or polyribosome. Okay, polysome or polyribosome. And this one is called mRNA. This is the mRNA. Single. Okay. Now in eukaryotic cell, give the heading as eukaryotic cell. Eukaryotic cell. Okay, eukaryotic cell. So eukaryotic cell are found in Protista, fungi, plants and animals. Protista, fungi, plants and animals. Okay. Again, in compartmentalization, present in eukaryotes. Okay, so compartmentalization means having compartments. This is a complex locomotory structure. There is complex locomotory structure and cytoskeleton. Cytoskeleton present in eukaryotes. These are present in eukaryotes. Cytoskeletons are present in eukaryotes. Okay. So what consists of the cytoskeleton? The microtubules, microfilaments, intermediate filaments, and intermediate filament. Okay. Microtubules, microfilaments, and intermediate filaments are the constituents of the cytoskeleton. Okay. Now make a table. I think making a table will do more. Make a table. Let me draw a table. All eukaryotic cells are not identical. So in case of plant cell, it is animal cell. In plant cell, the cell wall present, animal cell absent. Okay. In plant cell, plastid present, animal cell absent. Large vacuole present, absent. Centriole is absent in plant cell but present in animal cell. Okay. Now, now come to the next point, the plasma membrane. Plasma membrane. What is plasma membrane? Plasma membrane was discovered after the discovery of electron microscope. After the discovery of the electron microscope, the plasma membrane was discovered, because without the electron microscope, plasma membrane was not been able to be discovered because it was not seen by complex or compound microscope. So after the discovery of the electron microscope, plasma membrane was seen. So after 1952, plasma membrane was discovered. So discovered in 1952. Okay. Chemical nature of plasma membrane. Chemical nature of plasma membrane. What is chemical nature? RBC membrane organelle composed of lipid and protein. So RBC lipid and protein से ही बनी हुई है. Lipid is unsaturated fatty acid liquid in nature, and protein solid nature. Mosaic. Okay, the nature of plasma membrane. Nature of PM. PM doesn't mean Prime Minister. It's a short term for plasma membrane. Every time I have to write plasma membrane, I will write PM in short. Okay. The nature of plasma membrane. It's semi-fluid. Is semi-fluid. semi-fluid in nature or quasi-fluid. Quasi-fluid in nature. Okay. Fluidity of plasma membrane. Fluidity. Plasma membrane consists of protein and carbohydrate. Carbs. Protein P, carbohydrate C. Okay. Note down RBC 52% protein, 40% lipid and 58% carbohydrate. Okay, this is the constituent of RBC. RBC का constituent वही है. Now comes to lipid. What are lipid? Lipids are just fat. Lipids are the fat. Let me draw a structure first. Okay, this is the head. This is the head. This is the head, and these are the tail. So the head remains in the outside. Head are hydrophilic. That is, remains in outside. Tail are hydrophobic. Remains inside. Okay. Now inner side is protected by from H2O. Inner side is protected by H2O. Okay. Exchange of lipid from 1 and 2 within one layer. Okay. Lateral mostly they are on the lateral sides. Okay. Between 1 and 3, between two layers, flip-flop, rarely. Okay. Now draw a structure. Now see, I am drawing a structure. Just look at this structure. I will explain it. So I have marked it as 1, 2 and 3. So what is 1, 2 and 3 means? The 1 is the integral protein. Marked 1, integral protein. 2 is the peripheral protein. Periphery means in the outer side. Peripheral protein or external protein. Integral protein or internal protein. And what is the 3? 1, 2 तो हो गया, अब 3 का क्या है? The third is इंटीग्रल प्रोटीन का ही क्रॉस बाइलेयर है. इंटीग्रल प्रोटीन का ही क्रॉस्ड बाइलेयर है. Crossed bilayer of integral protein, that is transmembrane protein. Okay, now the plasma membrane has 3 states: living state, selectively permeable and transport. Main function. Okay, sugar, glycolipid. अगर लिपिड के साथ कोई शुगर है तो लिपिड के साथ कोई कार्बोहाइड्रेट या शुगर है तो वो ग्लाइकोलिपिड है. लिपिड प्लस शुगर. ग्लाइकोप्रोटीन, लिपिड प्लस प्रोटीन. आई एम गोइंग बाई एनसीईआरटी लाइन बाई लाइन. ओके, एनसीईआरटी के बाद हम कुछ नहीं पढ़ा रहे हैं, सिर्फ अभी तो एनसीईआरटी पढ़ा रहा है. इसके बाद अगर चैप्टर कंप्लीट हो जाए तो उसके बाद हम मेडिकल बूस्टर दे देंगे. ठीक, फिर तो ये नया चैनल है तो जितना ज़्यादा सब्सक्राइबर बनेगा उतने ज़्यादा ही हमको एनर्जी मिलेगा. If you like the content, please share it. It's my request. If you like the content, please share it, because a doctor is teaching junior doctors. Okay, I have never explained myself. I am a doctor, and I am trying to make underprivileged student to come into medical colleges. I'll never take any money from the YouTube students, and you can get all the contents on free and YouTube, and you can study this because these will only come in the NEET examination. And the more you practice, main thing to crack the examination is to practice. Just make practice and make your final examination a mock test. If you can make your final examination a mock test, you will crack it, and crack it in a good government medical college. Okay, transport are 3 types. Active transport, facilitated transport, and passive transport. So these are the 3 types of transport: active transport, facilitated transport, and what is called passive transport. Okay, in case of active transport, just write the points. No need to draw the pictures. It will not come. Just basic. You have to know the basics of everything, but not the drawing. Every drawing will not. It's not important in it. Active transport requires energy supplied as sodium potassium pump. Okay. Used in uphill process, facilitated transport, usage, carrier protein, and passive transport, no ATP required, downhill process, no ATP required, along the concentration gradient, okay. Seen in gaseous transport like O2, CO2 are generally seen in these transports. Okay. No carrier protein is required. No carrier protein is required. Okay. So for today, up to now, I will also make separate videos. I will start a physiology lectures very shortly. The botany will also continue, the physiology will also be continued, because reading botany every day doesn't look good for medicals, upcoming medicals. I know they will have for a physiology. So in physiology we'll start from the breathing and respiration in 2 to 3 days, and the botany will continue as it long. Okay, thank you, and do revise and memorize this, and please revise the starting lectures, that is lecture 1, lecture 2, lecture 3 daily, because you go on to continue with the new lectures. If you don't revise, you will start to forget the previous lectures. If you start to forget the previous lectures, that nothing will happen, because in near examination, but before examination, you will get so much load of physics, chemistry, biology, botany, zoology, organic chemistry, physical chemistry, inorganic chemistry, electrics, and everything will be messed up if you don't revise. So start revise daily. Okay, thank you.